Titanium dioxide, banned in Europe, is one of the most common food additives in the U.S. Written by Michaela Conley. Titanium dioxide is the most widely used whitening pigment in the world and has been linked to adverse health effects, particularly genotoxicity and intestinal inflammation. It is applied as food coloring and a whitening agent to a wide variety of foods, including chewing gum, cakes, candies, breads, and ice cream. Because of health risks, France banned titanium dioxide as a food additive in 2020. 2 years later, the European Union also banned titanium dioxide as a food additive. But in the US, titanium dioxide is found all over the grocery shelves. Candy like Skittles, Starburst, and Jell-O, gum like Trident White Peppermint Gum, and Mentos Fresh Mint Gum, cake products like Duncan Hines Creamy Vanilla Frosting, and Nabisco Chips Ahoy. Cookies are just a few of the myriad food items that contain the additive. A significant body of research, mostly from rodent models and in vitro studies, has linked titanium dioxide with health risks related to the gut, including intestinal inflammation, alterations to the gut microbiota, and more. It is classified by the International Agency for Research on Cancer, IARC, in Group 2 billion as possibly carcinogenic to humans. As a food additive, titanium dioxide and its nanoparticles in particular have been associated with DNA damage and cell mutations, which in turn have potential to cause cancer. When used as a food coloring, it is known as E171. With the rise of nanotechnology, research in recent years has also shown the dangers of titanium dioxide, TO2 nanoparticles, and their genotoxicity, which refers to a chemical agent's ability to harm or damage DNA in cells, thus potentially causing cancer. Nanoparticles. Over the last several years, nanoparticles have come under scrutiny for adverse health effects. Nanoparticles are ultrafine particles between 1 to 100 nanometers in diameter. To put this in perspective, the average human hair is around 80,000 nanometers thick. Because of their size, which can be engineered and manipulated at the atomic or molecular level, nanoparticles exhibit unique physical, chemical, and biological properties. Titanium dioxide is one of the most commonly produced nanoparticles in the world. Studies of titanium dioxide as a food additive suggest health dangers. Genotoxicity and cytotoxicity. Numerous studies have linked titanium dioxide to genotoxicity and cytotoxicity. Genotoxicity refers to a chemical's potential to cause DNA damage, which can, in turn, lead to cancer. Cytotoxicity is a general term that refers to a characteristic of being harmful to cells. French researchers studied how and where E171 nanoparticles enter the bloodstream, first studying the route through pigs and then in vitro with human buccal cells, for a 2023 study published in the journal Nanotoxicology. The research showed that the nanoparticles absorbed quickly through the mouth and then into the bloodstream before damaging DNA and hindering cell regeneration. In a 2016 study published in Scientifica, Cairo, Egyptian researchers examined the effects of titanium dioxide nanoparticles on the organs of mice by orally administering the food additive daily for 5 days. The results showed that the exposure produced mild to moderate changes in the cytoarchitecture of brain tissue in a time-dependent manner. Furthermore, COMET assay revealed the apoptotic DNA fragmentation, while PCR, SSCP pattern and direct sequencing showed point mutation of presenilin 1 gene at exon 5, gene linked to inherited forms of Alzheimer's disease. The researchers wrote, "From these findings, the present study concluded that TIO2 is genotoxic and mutagenic to brain tissue, which in turn might lead to Alzheimer's disease incidence." For research published in 2022 study in the journal Food and Chemical Toxicology, scientists examined the genotoxicity and the intracellular reactive oxygen species induction by physiologically relevant concentrations of 3 different TIO2 nanomaterials in CACO2 and HT29, MTX, E12 intestinal cells, while considering the potential influence of the digestion process in the NM's physiochemical characteristics. They found a DNA damaging effect dependent on the nanomaterial, along with the micronucleus assay suggesting effects on chromosomal integrity, an indicator of cancer risk, in the HT29 MTX E12 cells for all the tested TiO2 nanomaterials. Researchers concluded that the results showcase evidence of concern regarding titanium dioxide used as a food additive. In a study published in the journal Toxicology, researchers examined the effects of exposing human colon cancer cell line HEC 116 titanium dioxide food additives in vitro. In the absence of cytotoxicity, E171 was accumulated in the cells after 24 hours of exposure, increasing granularity and reactive oxygen species, inducing alterations in the molecular pattern of nucleic acids and lipids, and causing nuclei enlargement, DNA damage, and tubulin depolymerization, the scientists wrote. Researchers removed the additive from the culture, then examined the results 48 hours later. They found the removal of E171 was unable to revert the alterations found after 24 hours of exposure in colon cells. In conclusion, exposure to E171 causes alterations that cannot be reverted after 48 hours if E171 is removed from colon cells. A review published in 2022 in the journal NanoImpact evaluated the latest research related to genotoxic effects of titanium dioxide through in vivo studies and in vitro cell tests. Researchers summarized the results by stating TiO2 nanoparticles could induce genotoxicity prior to cytotoxicity and are likely to be genotoxic to humans. Inflammation of the intestines. Animal studies have shown that, when consumed as a food additive, titanium dioxide can induce intestinal inflammation. In a 2019 study published in the journal Nanotoxicology, researchers recreated the first phase of digestion in mice and fed them titanium dioxide, then examined whether accumulation occurred in the organs. Researchers wrote, significant accumulation of titanium was observed in the liver and intestine of E171 fed mice. In the latter, a threefold increase in the number of TiO2 particles was also measured. Titanium accumulation in the liver was associated with necroinflammatory foci containing tissue monocytes/macrophages. 3 days after the last dose, increased superoxide production and inflammation were observed in the stomach and intestine. Overall, this indicates that the risk for human health associated with dietary exposure to E171 needs to be carefully considered. A study published in the Journal of Agricultural and Food Chemistry in 2019 sought to examine the effects of titanium dioxide on intestinal inflammation. Researchers did this by feeding rats titanium dioxide nanoparticles and found that, after the course of 2 to 3 months, the animals had lower body weights and induced intestinal inflammation. The researchers also found the nanoparticles altered gut microbiota composition and aggravated chronic colitis. The rats also experienced reduced populations of CD4+ T cells, which are cells that help organize immune responses by prompting other immune cells to fight infection, regulatory T cells, and white blood cells in mesenteric lymph nodes. The researchers wrote, dietary TiO2 nanoparticles could interfere with the balance of the immune system and dynamic of gut microbiome, which may result in low-grade intestinal inflammation and aggravated immunological response to external stimulus, thus introducing potential health risk. For a mini-review published in the journal Particle and Fiber Technology in 2021, scientists wanted to evaluate whether TiO2 particles contributed to the development and/or exacerbation of irritable bowel disease, and whether they altered the 4 elements of intestinal barrier function: the intestinal microbiota, the immune system, the mucus layer, and the epithelium. The breakdown of these 4 elements can contribute to autoimmune, neurological, inflammatory, infectious, and metabolic diseases. Following the review, the researchers concluded data indicate that TiO2 is able to alter the 4 compartments of IBF and to induce a low-grade intestinal inflammation associated or not with pre-neoplastic lesions. Neurotoxicity. A 2025 paper published in Alzheimer's and Dementia provides prime evidence that nanoparticles of titanium dioxide used in food and cosmetics, and carbon black, found in rubber and pigments, bind to a specific receptor in neurons, corrupting its signaling and causing oxidative stress, inflammation, and overproduction of toxic amyloid beta peptides, which are key markers of Alzheimer's disease. This study sheds light on how exposure to these particles might increase the risk of neurodegenerative diseases and highlights the need for strategies to mitigate their impact on brain health. Scientists analyzed research that examined how titanium dioxide nanoparticles interact with the brain for a 2015 review published in Nanoscale Research Letters. The researchers wrote, once the TiO2 are translocated into the central nervous system through certain pathways, they may accumulate in the brain regions. For their slow elimination rates, those NPs could remain in the brain zones for a long period, and the Ti contents would gradually increase with repeated exposure. After reviewing dozens of studies, the scientists concluded long-term or chronic exposure to TiO2 nanoparticles could potentially lead to the gradually increased Ti contents in the brain, which may eventually induce impairments on the neurons and glial cells and lead to CNS dysfunction as a consequence. For research published in Archives of Toxicology in 2020, scientists fed one group of mice a solution containing titanium dioxide for 1 month and compared it to those that did not receive the additive. They found the richness and evenness of gut microbiota were remarkably decreased, and the gut microbial community compositions were significantly changed in the titanium dioxide group when compared with the control group. The tests also revealed that the titanium dioxide exposure could cause locomotor dysfunction or mobility issues by elevating the excitement of enteric neurons, which might spread to the brain via gut-brain communication by vagal pathway. The researchers concluded, these findings provide valuable insights into the novel mechanism of TiO2-NP-induced neurotoxicity. Understanding the microbiota gut-brain axis will provide the foundation for potential therapeutic or prevention approaches against TiO2-NP-induced gut and brain-related disorders. In a 2020 study published in the Journal of Trace Elements in Medicine and Biology, researchers conducted an in vitro experiment to analyze the effects of TiO2 nanoparticles on a human neuroblastoma, SY5Y cell line. The scientists evaluated reactive oxygen species, ROS, generation, apoptosis, cellular antioxidant response, endoplasmic reticulum stress, and autophagy. The results showed that exposure to the nanoparticles induced ROS generation in a dose-dependent manner, with values reaching up to 10-fold those of controls. NRF2 nuclear localization and autophagy also increased in a dose-dependent manner. Apoptosis increased by 4 to 10-fold compared to the control group, depending on the dose employed. Promotion of obesity-related metabolic disorders. For a review published in 2023 in the journal Environmental Pollution, researchers examined E171 as a possible factor promoting obesity-related metabolic disorders. Because gut microbiota play an important role in immune function maintenance and development, and because titanium dioxide as a food additive has been shown to alter gut microbiota, researchers wanted to review the dysregulations along the gut microbiota immune system axis after oral TiO2 exposure compared to those reported in obese or diabetic patients, and to highlight potential mechanisms by which foodborne TiO2 nanoparticles may increase the susceptibility to develop obesity-related metabolic disorders. The study authors discovered recurrent changes in the gut microbiota composition when exposed to titanium dioxide nanoparticles, with an imbalance of intestinal symbiotic microbiota. These changes and imbalances were also reported and played a role in the development of obesity, the authors wrote. This highlights foodborne TiO2 nanoparticles as an endocrine disruptor-like chemical promoting obesity-related disorders, the authors concluded. Colorectal tumors and preneoplastic lesions. In a study published in the journal Food and Chemical Toxicology in 2016, researchers investigated whether titanium dioxide exposure led to an increase in colorectal tumor creation in mice by using a colitis-associated cancer model. By measuring tumor progression markers, the researchers found that mice given titanium dioxide experience enhanced tumor formation in the distal colon. There was also a decrease of cells that act as a protective barrier in the colon. The researchers wrote, These results suggest that E171 could worsen pre-existent intestinal diseases. In a 2017 study published in Scientific Reports, researchers exposed rats to human-relevant levels of E171 to examine the effects of intestinal inflammation and carcinogenesis. They saw that a 100-day E171 treatment promoted colon microinflammation and initiated preneoplastic lesions, while also fostering the growth of aberrant crypt foci in a chemically induced carcinogenesis model. They continued, stimulation of immune cells isolated from Peyer's patches, which are clusters of lymphoid follicles found in the intestine, showed a decrease in Thelper, the-YFN, secretion, while splenic the-17 inflammatory responses sharply increased, researchers wrote. A 100-day titanium dioxide treatment promoted colon microinflammation and initiated preneoplastic lesions. The scientists concluded these data should be considered for risk assessments of the susceptibility to these 17-driven autoimmune diseases and to colorectal cancer in humans exposed to TiO2 from dietary sources. Alterations in gut microbiota. Research has shown that when ingested as a food additive, titanium dioxide and its nanoparticles can impact, alter, and slash or damage important protective bacteria in the gut, along with the metabolic pathways of gut bacteria. The tiniest particles of titanium dioxide can raise blood sugar levels and impair how the body processes glucose, among other health harms, according to new research in mice published May 2025 in Food and Chemical Toxicology. These findings raise important questions about the potential risks of titanium dioxide nanoparticles in food products, particularly in relation to metabolic disorders such as obesity and type 2 diabetes, the researchers say. Titanium dioxide nanoparticles appear to interfere with how the gut senses nutrients from food in the body and how it produces and releases key hormones involved in digestion and blood sugar level control. When the body can't control blood sugar properly, it can lead to type 2 diabetes and obesity by making it harder to use insulin and easier to store fat. Read more in Pamela Ferdinand's article for U.S. Right to Know. Tiny titanium dioxide particles in food raise blood sugar, disrupt gut hormones in mice, study finds. A 2023 study published in the journal Environmental Research, scientists examined the effect of titanium dioxide nanoparticles on important gut bacteria in mice. Their results show the growth inhibitory effects could be associated with cell membrane damage caused by titanium dioxide nanoparticles to the bacterial strains. Metabolomics analysis showed that TiO2-NPs caused alterations in multiple metabolic pathways of gut bacteria, such as tryptophan and arginine metabolism, which were demonstrated to play crucial roles in regulating gut and host health. The researchers also found that 4 different neuroprotective metabolites were significantly reduced in urine and in vitro bacteria and vivo urine samples. The researchers concluded, increasing evidence implies that the gut microbiome plays a profound role in regulating host metabolism. Our results illustrated that TiO2-NPs hindered the growth of 4 beneficial gut bacterial strains. Australian researchers examined how titanium dioxide as a food additive affected gut microbiota in mice by orally administering it in drinking water. The study, published in the journal Frontiers in Nutrition in 2019, found the treatment could alter the release of bacterial metabolites in vivo and affect the spatial distribution of commensal bacteria in vitro by promoting biofilm formation. We also found reduced expression of the colonic mucin 2 gene, a key component of the intestinal mucus layer, and increased expression of the beta-defensin gene, indicating that titanium dioxide significantly impacts gut homeostasis. The changes were then linked to colonic inflammation, along with a higher expression of inflammatory cytokines, which are signal proteins that help with regulation. The researchers concluded that titanium dioxide impairs gut homeostasis, which may in turn prime the host for disease development. In a small study published in the European Journal of Nutrition in 2020, researchers examined the effects of several food additives, including titanium dioxide, along with artificial sweeteners and cleaning products by testing the fecal samples of 13 people. Titanium dioxide was among the samples that induced significant shifts in microbiome community structure. The growth of the bacterium species belonging to C. leptum, which has been shown to decrease in patients with inflammatory bowel disease, significantly decreased in the presence of titanium dioxide, among other additives and sweeteners tested. In a study published in the journal Environmental Toxicology and Pharmacology in 2020, researchers examined the effects of food additives titanium dioxide and silica on the intestinal tract by grouping and feeding mice 3 different food-grade particles, micro-TO2, nano-TO2, and nano-CO2. With all 3 groups, researchers observed changes in the gut microbiota, particularly mucus-associated bacteria. Furthermore, all 3 groups experienced inflammatory damage to the intestine, but the nano-TO2 displayed the most pronounced changes. The researchers wrote, "Our results suggest that the toxic effects on the intestine were due to reduced intestinal mucus barrier function and an increase in metabolite lipopolysaccharides, which activated the expression of inflammatory factors downstream." In mice exposed to nano-TO2, the intestinal PKC/T LR4/NF signaling pathway was activated. These findings will raise awareness of toxicities associated with the use of food-grade TiO2 and SiCO2. Increased severity of ulcerative colitis. A 2023 study published in the journal Particle and Fiber Toxicology set out to examine the impact of titanium dioxide nanoparticles in mice on the course and prognosis of ulcerative colitis by creating an ulcerative colitis disease model. Researchers found that the titanium dioxide nanoparticles significantly increased the severity of colitis. They also decreased the body weight, increased the disease activity index and colonic mucosa damage index scores, shortened the colonic length, increased the inflammatory infiltration in the colon. Researchers concluded oral intake of TiO2 nanoparticles could affect the course of acute colitis in exacerbating the development of ulcerative colitis, prolonging the ulcerative colitis course and inhibiting ulcerative colitis recovery. Atherosclerosis. In a 2022 study published in the Journal of Hazardous Materials, scientists wanted to examine the effects of titanium dioxide as a food additive on atherosclerosis in mice. Atherosclerosis refers to a hardening of the arteries. Researchers fed mice 40 mg/kg of the food additive every day for 4 months and found that it not only altered gut microbiota, but also led to a significantly increased atherosclerotic lesion area, especially in animals that consumed a high choline Western diet, HCD. Promotion of non-cancerous tumors. Mexican researchers sought to evaluate the effects of E171 across a span of conditions in mice, including its influence on behavior, along with the effects on the colon and liver. The research published in 2020 in the journal Food and Chemical Toxicology showed that E171 promoted anxiety and induced adenomas, or non-cancerous tumors, in the colon. They also found that E171 heightened goblet cells hypertrophy and hyperplasia, which is typically seen in asthma patients and triggered by smoking or external pollutants and toxins. They also noted mucin's overexpression in the mice, which can be linked to cancer cell formation. Breathing problems in offspring. In a study published in 2022 in the journal Particle and Fiber Technology, researchers examined the impact of maternal exposure to titanium dioxide nanoparticles in newborn offspring mice. They found that a chronic exposure to TiO2 NPs during pregnancy alters the respiratory activity of offspring, characterized by an abnormally elevated rate of breathing. Breathing was also shown to be significantly and abnormally accelerated, and the ability for neural circuitry to effectively adjust breathing rates was impaired. The researchers concluded, "Our findings thus demonstrate that a maternal exposure to TiO2 NPs during pregnancy affects the normal development and operation of the respiratory centers in progeny." Decreased vitamin D bioaccessibility. In a 2021, Chinese researchers examined the impact of E171 on lipid digestion and vitamin D3 bioaccessibility in a simulated human gastrointestinal tract model. They examined vitamin D's bioaccessibility, or the amount it was released in the gastrointestinal tract, becoming available for absorption, and found it significantly decreased from 80% to 74% with the addition of E171. In the experiment, E171 decreased lipid digestion dose dependently. Researchers wrote, "The findings of this study enhance our understanding toward the potential impact of E171 on the nutritional attributes of foods for human digestion health." The study was published in the Journal of Agricultural and Food Chemistry. Exposure to titanium dioxide in utero and in breastfeeding children. In a review published in 2022 in the Journal Archives of Toxicology, researchers found that the ingestion of E171 is a definite health risk for consumers and their progeny. After reviewing dozens of in vivo, ex vivo, and in vitro studies on the toxicity of E171, the researchers wrote that two facts must be noted. First, reprotoxicity studies show that animals of both sexes are impacted by the toxicity of these nanoparticles, underlining the importance of conducting in vivo studies using both male and female animals. Second, human exposure begins in utero via maternal fetal transfer and continues after birth by breastfeeding. Children are then chronically re-exposed due to their food preferences. To be relevant to the human in vivo situation, experimental studies should therefore consider nanoparticle exposure with respect to the age or life period of the study population. What exactly is titanium dioxide? Titanium dioxide, TiO2, is a chemically inert inorganic compound and an insoluble white solid that occurs naturally in several minerals, including rutile, anatase, and brochite. It is created synthetically from the mineral ilmenite. It is an insoluble white solid. Anatase, when compared to brochite and ruthenium, has the most industrial applications, but it is the most toxic form of TiO2. There are numerous manufacturers of titanium dioxide. The largest include Delaware-based Chemours, a spin-off of DuPont Chemical, Texas-based Pronos, and China-based Lohmann Billions Group, all of which manufacture pigments for use in products like paints, coatings, and plastics. UK-based Venator is a major supplier of titanium dioxide used in food and cosmetics, along with paints, paper, plastic, and more. As a pigment, it is called pigment white 6, PW6, titanium white, or CI77891. As a food additive, it is known as E171. Nanotoxicology. Nanotoxicology focuses on determining the adverse effects of nanomaterials on human health and the environment. In general, nanoparticles have been shown to accumulate in the body, particularly in organs in the gastrointestinal tract, along with the liver, spleen, and capillaries of the lungs. Titanium dioxide nanoparticles are commonly found in a wide range of consumer goods, including cosmetics, sunscreens, paints and colorings, ceramics, glass, textiles, construction materials, medicine, food packaging, and more. In Europe, cosmetic companies are required to label products that contain nanoparticles. In the US, companies are not. In 2017, French researchers from the Institut National de la Recherche Agronomique, INRAE, were among the first to examine the effects of E171 nanoparticles on the body. They fed rats a dose of 10 milligrams of E171 per kilogram of body weight per day, which was similar to human exposure in food. The research, which was published in Scientific Reports, showed that E171 was able to traverse the intestinal barrier, pass into the bloodstream, and reach other areas of the body in rats. Researchers also found a link between immune system disorders and the absorption of titanium dioxide nanoparticles. Titanium dioxide nanoparticles have also been found in human placenta and in infant meconium, indicating its ability to be transferred from mother to fetus. A 2012 study published in the journal Environmental Science and Technology noted that children are especially exposed to titanium dioxide because of the food that contains the food additive and is particularly marketed to children, including candy and cakes. Recent policy changes in regard to titanium dioxide. EU ban on titanium dioxide. The European Commission banned titanium dioxide as a food additive in the EU in 2022 after the European Food Safety Authority, EFSA, conducted an updated safety assessment of E171 and concluded the panel could not eliminate concerns about its genotoxicity. EFSA scientific conclusion on E171. Following a request for assessment in 2020 by the EU, the European Food Safety Authority, EFSA, assessed E171, particularly for its genotoxicity. In 2022, the agency deemed the food additive no longer safe for use. Professor Maj Younes, chair of the FSA's expert panel on food additives and flavorings, FAF, wrote of the decision, taking into account all available scientific studies and data, the panel concluded that titanium dioxide can no longer be considered safe as a food additive. A critical element in reaching this conclusion is that we could not exclude genotoxicity concerns after consumption of titanium dioxide particles. After oral ingestion, the absorption of titanium dioxide particles is low, however, they can accumulate in the body. Professor Matthew Wright, chair of the FSA's working group on E171, noted, although the evidence for general toxic effects was not conclusive, on the basis of the new data and strengthened methods, we could not rule out a concern for genotoxicity, and consequently, we could not establish a safe level for daily intake of the food additive. FDA's response. Following the EU's ban on E171, the Food and Drug Administration, FDA, told The Guardian that, based on current evidence, titanium dioxide as a food additive is safe. The available safety studies do not demonstrate safety concerns connected to the use of titanium dioxide as a color additive. Currently, titanium dioxide as a food additive is classified as GRAS, or generally recognized as safe. The FDA has not updated its general guidance on safety assessments since 2007. Within that time, there has been a significant increase in research on the confluence of toxicology, nanotechnology, and human health. The EU updates its guidance regularly with new science available to offer proper safety assessments, with its most recent update published in 2021. As for titanium dioxide, the FDA approved titanium dioxide for use as a food additive in 1966. The last time the agency reviewed the additive safety, according to The Guardian, was in 1973. States looking to ban titanium dioxide as a food additive. In 2023, California and New York proposed banning several food additives that are banned in Europe but legal in the United States. Titanium dioxide was among the 5 proposed to be banned, but in September, the additive was removed from the list of additives from the California ban list. Australia and New Zealand review of TiO2. In 2022, a year after the EFSA recommended against the use of E171, the Food Standards Australia-New Zealand, FSANZ, conducted its own reassessment of titanium dioxide as a food additive. The agency concluded that titanium dioxide was indeed safe to use as a food additive. The United Kingdom and Canada came to similar conclusions. Journalism. Tiny titanium dioxide particles in food raise blood sugar, disrupt gut hormones in mice, study finds, by Pamela Ferdinand, U.S. Right to Know, May 12, 2025. Why is a sunscreen ingredient in my frozen pizza? By Andrea Peterson, Wall Street Journal, June 10, 2024. Food additive nanoparticles could negatively affect your gut health, by Gillian McCarthy, Binghamton University, May 4, 2023. Food additive or carcinogen. The growing list of chemicals banned in EU but used in US, by Michaela Conley, Guardian, June 23, 2022. Public health groups urge FDA to cancel titanium dioxide in food, by Center for Science in the Public Interest, May 30, 2023. Omen bans food products containing titanium dioxide, by Gulf News Report, July 23, 2023. Titanium dioxide, E171, no longer considered safe when used as a food additive by European Food Safety Authority, May 6, 2021. Health advocates urge U.S. regulators to ban common food coloring additive, by Tom Perkins, Guardian, June 2, 2023. A lawsuit claims Skittles are unfit for consumption. Experts weigh in, by Rachel Rabkin Peachman, New York Times, July 26, 2022. The Trouble with Titanium Dioxide by Siloam Springs Regional Hospital, Herald Leader, October 4, 2023.